So, when we teach our students of this in the main, you have a cell here, maybe a stem cell, and goes mutation selection, and a tumor suppressor gene or oncogene. You get a benign tumor, and then you get further mutations, and you get a primary cancer. That's the orthodox view. But if you embrace the slaughter hypothesis, what it's saying, in fact, is that you're getting a progenitor cell or stem cell down here. And then either through genetic or epigenetic changes, you get an expansion in that stem cell number, which is genetically altered. And this stem cell, mutated stem cell, whatever, spreads through the epithelium and is available to undergo further changes. And it's only by postulating this sort of change that you can really account for the frequency of cancer, because somatic mutations in humans are really quite rare. But if you have a large field effect, let's say right through the man's colon, for example, then you can explain why he gets so many tumors. So that's really what we're talking about. So in the gut, we're really sport for choice about looking at these phenomena. There's a very nice example of this lovely disease, Barts esophagus, which you're all familiar with, where you get replacement disease, distended esophagus with mucin-secreting epithelium exposed in crypts, and the work of Brian Reed in Seattle has shown, fairly conclusive in their view, that you actually get the spread of the same mutation all the way through the mucosa. And that's regarded as classical work until recently. Then, of course, you have intestinal metaplasia in the stomach, which is thought to be the forerunner of gastric carcinoma. You get change from the gastric phenotype, the intestinal phenotype. And you see down here there's a patch taken with an endoscope, and so this spreads as well. Then you have the archetypal example of ulcerative colitis, where you get indolent ulcerative colitis, chronic ulcerative colitis, eventually goes on to give you carcinoma and dysplasia. And then, of course, the prototypical example is the adenoma, where you get, you can see here there's a crypt, then you get this monocryptal adenoma, which is the first adenoma you can see there's a crypt there which is occupied by transformed cells, and that eventually goes on to give you an adenoma. So it spreads. So how is all this spreading going on, and how do you get fixation in these things? Well, I'm sure you know most about this. This is just a tissue stain with P67 to show you where the cryptic cells are. The small intestine, they're down at the base of the crypt here. Down here, there's the girth, and then the colon very much the same. The lower half of the cryptid, or where the proliferating cells are found, they migrate up and fall off the end after they differentiate and then work. And what we think is happening is that all stem cells in the gut are enclosed in a thing called the niche, stem cell niche. And in the small intestine here, it's around about here, just around the Panner cells, either between the Panner cells or just above them. And stem cell niches are very important, because it's not just the stem cells that are important, it's these surrounding cells, these so-called niche cells, the myofibroblasts. And these are the cells that in the gut are the source of the Wnt ligand, and the frizzled receptors are on the stem cells. And there's a lot of crosstalk between the mesenchyme and the epithelial cells, which maintain the stem cell niche. And it's highly likely that these cells maintain the symmetry of stem cell division. And we subscribe in the gut to this what's called the Unitarian hypothesis of Charles LeBlanc, where there's a stem cell in the base which gives rise to all cell lineages, the Panner cells, the Gothic cells, the endocrine cells, and the enterocytes by a series of committed progenitors. So I suppose what the idea is that you're going to have stem cell in this niche down here, and this stem cell is going to acquire a mutation, and then that's going to spread throughout the niche and gradually expand. So the question I want to ask, is it possible in human beings to look into that stem cell niche and try and look at stem cell dynamics, why this is happening? And I hope to convince you we can. So, where's the handle? Well, it really came through talking to a very good friend of mine in Newcastle, Turnbull, who is interested in mitochondrial DNA mutations. This unfortunate lady has this complex syndrome where she's got an inherent mutation in the gene which encodes for transfer RNA to lysine, which is a mitochondrial gene. And she has all these unfortunate stigmata. And we see how this is inherited. And when you take muscle biopsies from these people, you can see you can pick out very easily the mutated muscle fiber. This is stained for an enzyme called cytochrome C oxidase, which is part of respiratory complex 4. And you can see the wild type fibers are brown, that they're wild type, and the other ones don't stain. If you stain for a nuclear encoded enzyme like succinate dehydrogenase, you can pick these mutated fibers out in blue. And that's very nice. You can see how. So what Dr. Turnbull was thinking about was that these are mainly post-mitotic cells. But can you use this to actually look at stem cells and their biology? And I'm trying to convince you can. So this is the technique. It's comparatively simple. It's done in frozen sections. It's just double enzyme dislocation. And so you take cytochrome deoxidase, which is encoded entirely by the mitochondrial genome. Yes? You stain, and the reaction product is brown. And then to pick up any mutations, you stain for succinate dehydrogenase, perhaps cycle you remember, and that's encoded entirely by the nuclear DNA. So it's not affected by mitochondrial mutations at all. And the sort of pictures you will see in all your young people, you see that you find no crypts which are at all mutated. They're all brown, they're all wild type, which is very nice. But as you get to my age particularly, and Professor Williams' age, you can see lovely pictures like this. Now I think this really turns me on. Never turns you on. You can see here that there is a possibly orgasmic mutated crypt. Yes? It's the first human mutated crypt I've ever seen. I've seen plenty of rats and mice when you give them genotoxic carcinogens. But you see here, this crypt is entirely mutated. There's no brown staining at all. So what's happened here? The mutations appeared in the cytochrome deoxidase gene in a stem cell, down here. And that's taken over the stem cell niche, so-called niche succession, and then taken over the entire crypt. Because this is a new clone, it's called monoclonal conversion. So that's very exciting. But you have to show this is really mutated, and that's not so complex as you think. Well, all you do is do laser microdissection of all parts of this crypt. You can PCR the entire mitochondrial genome, there's only 27, 28 genes, and you can then look at the entire mitochondrial genome. You can see these cells here got a stop codon in cytochrome deoxidase, which stops transcription here. And so all these cells are mutated, so you know profoundly the crypt. And you can take it even further, because you can then do serial section. And here's serial section, and you can then build a model. Remember, the blue cells are mutated cells. And you see, here's the ribbon. It starts in the base here and runs all the way up the side of the crypt. So there's a sort of clone expanding within the crypt. And of course, because the origin of the flux is here, we can say the stem cell niche is down here in the base. But we knew that from animal studies. But this method is now being applied to the pancreas and the liver and other tissues where we don't know what the niche is, and has the potential for showing where stem cell niches are in human beings, which is the only method I know. So what I think is happening here is that here's your stem cells in the base of the crypt. One of these cells accumulates sufficient numbers of mutant cytochrome deoxidase molecules into mitochondria. Remember, there are 800 to 900 of these organelles in each cell. It takes quite a long time. And at the age of 40, you start getting this phenomenon seeing it. Then that probably by some sort of stochastic method, because this does not confer any advantage. This cell then takes over the entire niche here. And so what you find here, there's a mutant cell there in the base of the crypt. This then expands to form half the crypt, which we saw, and gradually the whole thing becomes taken over. So you've got expansion of the clone, you've got niche succession, and monoclonal conversion. So you're now getting a mutant stem cell taking over the entire crypt, which is very, very nice. But just taking it further, how does this mutation then expand?